So in the last unit, we looked at cells, and we knew that a cell membrane is a really important part of a cell because it's I forgot the terminology. Let me look at it. Selectively or selectively permeable, semi-permeable. It only allows certain things in and certain things out. That's really important because there's a lot of internal components to a cell. You got to keep all of that packaged. That's what the point of a cell membrane is, right? Cell membranes are made of this bilayer of phospholipids. So right here are these phospholipids. You hear you hear lipid, and you're probably thinking lipid, fat, non-polar. Lipids are non-polar. But what's interesting about a phospholipid is because of its, chemical, because of how it's made chemically, not made, I don't know why I'm saying it like that. Because of the chemical makeup of a phospholipid, it has a non-polar and a polar part to it, and we call that, let me look at the term, I always forget these terminologies, amphiphatic. Amphiphatic just means, hey, it has a hydrophilic head right here, right, this head right here, so I can write, head is hydrophilic, but the tail is hydrophobic, and that makes sense that the lipid bilayer looks like this because the polar head, right, the hydrophilic head, right, things that are polar, like water, they are hydrophilic, they're going to be surrounded on the outside. So if this is the inside, and in the inside, the cytoplasm has water, outside, you're in whatever organ or tissue you're in, there's probably going to be some water. The head is going to be on the outside. Meanwhile, the hydrophobic tail, which is non-polar, is going to be out on the inside. Now, another part to the cell membrane, I actually haven't drawn them, but we have these pieces of cholesterol. So here's the deal. We call this model of the cell membrane, we call this the fluid mosaic model. Fluid meaning that it's fluid in the sense that it moves around. It's not like a tight structure. And mosaic, because it's this mosaic of all these, all the parts that make it up, right? It's this mosaic, right? A mosaic is just a bunch of small things creating a larger picture. The fluid mosaic model, right? What keeps this thing fluid is, well, the lipids, these lipid molecules, they're moving around, so they have that fluidity. The cholesterols help that because when the temperatures get really low, these molecules, they want to pack together. But this cholesterol, right here, I'll just say, there we go, cholesterol, it kind of acts like a stopper. It stops them from fully getting packaged. So that can be pretty important to keep that fluidity. Meanwhile, if the temperatures get really hot, these molecules are probably moving around a lot. The cholesterol helps keep it together. So the cholesterol just helps kind of maintain that phospholipid bilayer structure. And the other thing we have are these big glob thingies right here. So these are proteins, and this one that I just labeled right here is what we call an integral protein. It's integrated through the bilayer. That's kind of where the name comes from. Integral, like it's integrated through, I guess. I don't even know if that's where the name comes from, but that's what makes sense to me. And integral proteins, they mostly help, for the most part, they mostly help with transporting molecules through. Here's the deal. Oxygen and carbon dioxide, those are like the reactants and products. I don't really fully understand cellular respiration and all that just yet, but they're involved in breathing and cellular respiration. So it would make sense that oxygen and carbon dioxide can pretty freely pass through this bilayer, right? And the reason that it can is because both of these are non-polar and they're pretty small. Non-polar is important because, hey, inside of here we have a hydrophilic tail kind of on the inside. That's going to not be a fan of polar. And if anything's non-polar, if anything's like an ion, it's just not going to pass through because of that chemistry, right? Think about think about oil and in water. They don't mix, right? So that's important for CO2 and oxygen to be nonpolar. That allows them to pass through pretty freely with diffusion, right? And we'll talk about diffusion, but this is what we call simple diffusion. They're just able to simply pass through the cell membrane. But for something like water is, it's, and these molecules have to be relatively small. Water's not really large, but it is polar, so it's gonna have an issue going through the hydrophobic part of the cell membrane. So it's really important that this integral protein is here, because the integral protein, it allows the water to go through this channel. And specifically in this case, we would call this an aquaporin, right? Aquaporin is this integral protein, what we actually call a carrier protein, channel protein, that allows the water to go through. And, boom, we get osmosis, we get the diffusion of water. That's what osmosis is. So integral proteins, you can have a channel protein, right? So a channel protein is like something like this, like an aqueduct, I guess. It's able to slide through, pass through the protein, and it's going to be hydrophilic, right? It has to be hydrophilic for stuff like water, other polar things to go through. And the other kind of integral protein is a channel protein. So channel proteins, I need to look through more further. Basically, if we had, let's say this was our, bilayer, right? This is our little bilayer. I'm just really roughly sketching this. And you had a molecule trying to go through. It kind of sits through here, and then the carrier protein kind of allows it to go through, and it kind of transports it through. I need to kind of understand that more, but the idea is that carrier proteins will carry it through. Did I write channel twice? I meant carrier. Carrier protein carries through an object, which is different from endocytosis, because endocytosis is a form of active transport. This is a form of facilitated diffusion. It's still diffusion. There's no energy required for water to go through this channel protein. It's just following the concentration gradient. And we'll talk more about that in the next video. I just want to give you an idea of what is a cell membrane made up of. We have these peripheral proteins right here. I'm not going to add too many sketches now. I might as well. These peripheral proteins are these big globs, and peripheral kind of means on the outside, right? It's kind of lodged in on the kind of outside of the cell membrane. It does a bunch of tasks. It can act kind of like an enzyme, they can do enzyme stuff and probably facilitate reactions like how an enzyme would. It also attaches to the cytoskeleton, and that helps maintain the shape of the cell. The main thing not the main thing, because these peripheral proteins can have a lot of functions, but one kind of thing is you can have a carbohydrate attaching to this peripheral protein. We call that a glycoprotein, and a glycoprotein is important for cell-to-cell recognition, right? Identifying, hey, on the outside, is that a cell or is that something else? A pathogen. So you can imagine how a glycoprotein could be really important. And so this thing right here is a carbohydrate. If a carbohydrate attaches to a lipid, we get a glycolipid, which I'm not 100% sure what the function of a glycolipid is. I have to look into that. But once again, just showing you what is a cell membrane made up of. That is basically the main components. Let's see if I missed anything from my notes. I don't think I really did. I think this is a cell membrane. So we'll talk more about how the cell membrane does the different types of cell transport, and we'll talk about diffusion, talk about all of that. So thank you for watching this little lesson.